So, good morning. Many of the points I'd like to discuss with you were touched upon during the early discussion, but I'll try to expand on them a little bit. And I'll start by mentioning this Science for Life laboratory, which Gunnar and I are vice heads of for Stockholm and Uppsala, respectively. Part of the idea with that is to make large-scale biology tools available to all scientists in Sweden and also to the industry in Sweden. And we talk both about more standard techniques, and an example of that would be next-generation sequencing, which I think many of you are aware of has undergone a revolution and picked up speed fabulously, but also more original tools that we think we are particularly well-equipped to do and now try to bring to applications more efficiently than otherwise. And the tools I'll be talking about are examples of things that we now hope will be more generally available, and we'll be able to reap the fruit of them. So, starting off, I'll be talking about protein biomarkers, which I think is a really exciting area. And why do I say it's exciting? Well, I've listed some of the reasons, and the one I put first is the one that I think is the really most important one, namely the potential of diagnosing any disease anywhere in the body with a simple blood sample at an early stage before the patient sustains significant tissue damage when it's still time to repair. In medicine, we're quite successful curing disease if you catch them early, and we're quite unsuccessful late in the phase. Maybe regenerative medicine will change that, and we will be able to repair even very seriously damaged organs, but currently it's really very important. So the possibility, even if it's just a glimmer of a hope of diagnosing diseases at an early stage, I think it's really worth putting effort into, and that's what we're doing. And I'll try to convince you that we may have some chance to make headway there. Second in line, still extremely important, is the possibility of finding what drug goes with what patient. And that is brought about and becoming a necessity, partially because of this revolution in biologics that Lotta will be telling you about. These drugs are very expensive, and it's not meaningful to give an expensive antibody therapy to a patient who does not express the protein that the antibody is targeting. So with these drugs, it's really important to have what we call companion diagnostics or theranostics, these terms that are being used, or RxDX, many terms for this. But I think that will bring with it the notion that we increasingly any drug will have to be associated with a diagnostic. And this is going to be true during drug development, and companies like Roche at least claim that when a new drug enters a pipeline, it's associated with a new diagnostic, which will follow it all the way into the routine diagnostic care. And finally, finding out how patients are doing, for instance, after therapy. In that regard, a very important area, and perhaps commercially the biggest part of diagnostics will be in monitoring healthcare at the point of care. That is, not central laboratories running large, slow tests, but the doctor just taking a sample and quickly telling you what's wrong, or maybe even you yourself in the kitchen running a test to finding out, do I have a bacterial or a viral infection? That's likely to become very important. So all these things are reasons that things will develop. And one further reason, which also was touched upon before, is the fact that we are now building across Europe research infrastructures that will help work of this nature. And I just mentioned one which I am associated with called BBMRI, Biobanking and Biomolecular Resource Research Infrastructure, which is an effort to coordinate the collection of patient samples across Europe. This is an area where we in Scandinavia pride ourselves that we have been at it for a long time, and our population has been quite agreeable to provide samples, but it's now being built across Europe, and the way it's organized is that we are collecting. Some people are worrying about population-based biobanks. So in Sweden we have a couple of such programs. You have them in England also, where we just pick samples from anybody as whatever their condition is. We also have more disease-oriented biobanks. And that goes along with also patient records. In Sweden, we have a very efficient way of collecting information, both about the outcome. We know everybody who's been treated at the hospital for any given diagnosis, and that can be connected to the patient samples. But then we also have the work package on technologies and reagent resources. And this is a work package that I am coordinating together with a British colleague, Mike Taussig, in Cambridge. And of course, this is very much my interest and very much the purpose of SciLifeLab also, to make sure that the best tools are available for these studies and to sort of bring up the rear, make sure that nobody's using old-fashioned technology and wasting samples. We also have a work package on databases, and again, this work package is also coordinated out of Sweden, Stockholm, Jan-Erik Litton, and then there's a work package on ethics and legal issues, which is also a very important point. So all these are reasons that one can be optimistic, but things are not going so well, as I already said. There's a very slow trickle of new biomarkers entering the market. The red spots here demonstrate markers. And what this shows you is a concentration range of biomarkers. So some things are very abundant. Serum albumin is the #1 protein, the most abundant. And from then on, it goes down till you hit something like tumor necrosis factor alpha. And then you've gone through 10 orders of magnitude. And it's really important to try to get that into your head, that there is a tremendous complexity of proteins. And if you're looking for something down here, then there is a lot of other things that you have to ignore. Part of the problem I think we've been facing is that we have used completely insufficient tools that have no chance of finding the rare things. So I think there are 3 areas that are going to be really important going forward. One is to build more specificity in how we look into protein, and I think we've completely shambled that so far. We haven't used the right tools. We also need to be able to look at more things in parallel, because we can't afford to take one assay per protein per patient sample. We want to look at large numbers of protein. And in many cases, as Gunnar touched upon before, proteins don't act alone. We want to know constellations of protein, how they are working together. That will be a new class of biomarkers, and I'll show you one example of that. And here I become a little technical, and some of you may not care for this so much as I do, but I'll just very superficially describe this to you. This is a standard approach which is used extensively in many groups, and a lot of money is put into approaches like this, where you use something like an antibody directed to a protein. You put it in a little spot, maybe along with many other spots with other antibodies, and then you label your sample. You put a fluorophore or something on all proteins in your sample, and you see where do they end up, which antibodies bound them. And that will tell you something, but antibodies are not to be trusted alone. There's a lot of work to get the therapeutic antibody, to find the ones that have the right specificity, and even they have some cross-reactivity. We are using something similar to how PCR changed the situation for DNA analysis. PCR requires 2 recognition events to get a signal. We are building assays where you need 2 or 3 different antibodies to recognize the same protein. And the way we do that is to attach DNA strands onto the antibodies. Again, I'm being very technical here, but if these 2 antibodies can bind this protein, then these DNA strands are brought in proximity and can be joined by something we call proximity ligation assays. And this is a technology that's held by a company we spun out called Olink Bioscience, O-L-I-N-K. And that means that now we're back in the world of DNA. We don't have to worry about the difficult proteins. Everything is just standard DNA. So we can now just read this off, and we encode the DNA with information that tells us what protein was being detected. And as you know, there's been a revolution in DNA sequencing. We can now sequence 1 billion molecules in parallel. So we can take these little pieces of DNA, each of which represent one detected protein, and we can put on that a little piece of information that tells us what was the sample. So we can take samples from 100 patients, look at many proteins, and then just sequence them through to identify them. How many times did we see this protein in this patient? This is a rather unique technology, and don't worry about the details in this slide, but this is just an example of such an analysis where we looked at the number of proteins in a patient with cardiovascular disease. So it's a very well-controlled study. Patients present at the hospital with chest pain. Some of them turn out to have a myocardial infarction, some of them don't. The ones that didn't are blue, and the ones that did are red. And we've just plotted their levels. And you can see in cystatin B, there is a difference between the red line, each dot is one patient, and the blue line, one control. Whereas for other things like cystatin C, there is no difference. Cystatin C is not a marker. You can't tell if a patient has myocardial infarction, but you can get some information on cystatin B. However, there are some normals that have higher levels than some of the patients. So by itself, it's not going to do it. And I think that's a pattern. We will have to use many different markers. But this is a rather powerful technology which has the advantages of specificity, ability to multiplex, looking at many things in parallel, and as I'll show you also, the ability to look at more complex targets. So maybe I'll, before I get into that, to end off, I think biomarkers will look very different. We will start looking for other things.